So, after the last presentation, I would like to present the evidence in favor of a lab origin. So, the evidence in favor of a lab origin mainly comes from the records of the planned, proposed, and funded research at the Wuhan Institute of Virology, undertaken mostly in collaboration with U.S. research group EcoHealth Alliance and funded in part by the NIH. The two principal grants that I would like to focus on is understanding the risk of bat coronavirus emergence, which was funded from 2014 to 2019. And keep in mind this was funded. And the other grant proposal is Project Diffuse, proposed in 2018, which, as far as we know, was not funded but falls within the scope of the previous grant, the Understanding Grant, which was funded. And I'll talk about these two proposals in much more detail. Now, the first point, our piece of evidence in favor of a lab origin, is the geographic coincidence between the closest relatives of SARS-CoV-2, between where these closest relatives are found, and where the Wuhan Institute of Virology sampled. We know they sampled in Yunnan province and Laos. In the Yunnan province and Laos in Yunnan we find RATG13, and in Laos we find the banal viruses. And the four closest relatives of SARS-CoV-2 have been found here and here, which is right where the Wuhan Institute of Virology was sampling. And I'll get more into RATG13 later, because there's some suspicious stuff there. But it's undisputable that the closest relatives of SARS-CoV-2 are found in Yunnan and Laos, and that's where the Wuhan Institute of Virology was sampling. So that's a piece of evidence. Where are the closest relatives? Where they were sampling. And again, this is from the Understanding Grant Report. Yunnan, Mojiang specifically, because we'll come back to that. Yunnan, Mojiang, Laos. So we know that they were sampling where the relatives were found, and we also know they were conducting spillover studies on novel SARS coronaviruses, right where a novel SARS coronavirus spilled over. Now, here we have the grant description from the funded grant, Understanding the Risk of Bat Coronavirus Emergence. And here they're saying emergence potential will be tested experimentally using, among other things, virus infection experiments in humanized mice and cell culture. So this is live virus they plan on using. So they're going to test their predictions of emergence potential and interspecies transmission risk using live virus. And notably in 2019, they changed their aims, they changed the wording. It's still more or less the same. And here they mention characterization of spillover risk to identify viruses of public health concern using infectious clones, in vitro and in vivo infection experiments to test the hypothesis that divergence thresholds in S protein sequence predict spillover potential. And if you look more into the grant, in the actual text of the grant, they mention 10 to 25 divergence in this S protein sequence. I assume this is amino acid sequence. That's the only thing that makes sense. And so first, this is a coincidence that, again, evidence showing spillover studies were being conducted where a virus of that type spilled over. And now we're going to look into this S protein sequence data. Because as we know, as I just showed, they were looking for spikes that were 10 to 25% divergent. What does the evidence show? The evidence shows that SARS coronavirus 2 spike is 24% divergent. So it fits right within the range that they were looking for. So again, this is a virus that fits their divergence criteria that they were looking for where its relatives were found. This is already two coincidences. And they were actively seeking viruses with this level of divergence. They say actively seek, and this is the text they added in their 2018 grant report. So as of the end of 2018, early 2019, they were actively seeking viruses that were this much divergent. And it's interesting to note these numbers here for HKU3 is not correct. So the question is, why were they looking for 25% divergent SARS coronaviruses? Because HKU3 coronavirus is not 25% divergence. This is, at a minimum, an error. But they're claiming the ability to bind ACE2 is lost when the S protein divergence is between 10 and 25%, although no viruses within this range have been described. This is just not true. If you align HKU3 with SARS-CoV-1, because that's what they were referring to at the time, it was less than 22% divergent. So where are they getting the 25% number? I don't know, but that's suspicious to me. In the interview with Congress, Ralph Barrick described a completely different way that they arrived at their 25% number, where he says they have 30 to 35% variation. And as I just showed, they have less than 22% variation. And he describes a method where you take 30 to 35 and basically take midway between the 10% divergence that they know still works, and you get 20 to 25. And this is it's strange to me that both explanations are not the same, and they both use incorrect numbers for HKU3 divergence. And yet somehow they arrived at a number that almost perfectly matches that of SARS-CoV-2. It makes me wonder if they already had some viruses in mind and wanted to look smart when they validated their predictions. And on top of this, the explanation is just nonsensical, because we know NL63, which is another coronavirus, uses ACE2. This is another coronavirus that infects humans, and its amino acid sequence is 70% divergent, over 70%. So we know it can use ACE2 even if it's 70% divergent. They knew ones that were 22% divergent didn't infect human, or didn't use human ACE2. So where they get this 25% number for the ACE2 threshold is beyond me. It does not make any sense. And yet SARS coronavirus 2 matched the number they had in their grant reports. Now, it's important to note that they wanted to test emergence potential experimentally and test their predictions of interspecies transmission. And they had previously predicted that furin cleavage sites were critical for transmission of MERS to humans. And here when they added a furin cleavage site by mutation of HKU4, they were able to get it to infect humans. So we know that furin cleavage was one of these predictions they had for emergence potential. And the testing of a furin cleavage site would fall within the scope of their AIM-3 in this funded grant. So we know that. So when I hear diffuse wasn't funded, the idea of testing the effect of a furin cleavage site is still within the scope of this grant. Now, diffuse, as I said, mentions adding a furin cleavage site. And it's strange to me that they're going to analyze SARS-CoV-2 gene sequences for the presence of furin cleavage sites, because none had been found at this point. and there was no reason to think they would be found. So some people think this means they probably had already found some and just wanted to look smart by having their predictions validated. But regardless, they were looking for furin cleavage sites, and they wanted to introduce human-specific cleavage sites and evaluate growth when they were not present. Here they describe mismatches. And if you look in their grant proposal, it's not clear what they consider a mismatch, but here they have an alignment where you have this gap. To me, that could qualify as a mismatch. And they also wanted to look at low risk or deep sequencing data, which comes from cell culture or cultured virus, and look for any proteolytic cleavage sites, which includes furin cleavage sites, and if so, introduce these into a parental strain. This is not pseudovirus they're talking about. Now, it's interesting to note they mention positions 757 and 900 of S2 in other coves, which we have marked here, furin site S1, S2. So those that tell you that Diffuse didn't discuss S1, S2 or didn't propose furin cleavage site addition, that's simply not true. And the interview with Ralph Barrick also confirms this. In the interview, they asked him whose idea it was. He says the idea of manipulating the protease was clearly his, no question. And he's also mentioning he's specifically interested in FIPV, where when the furin cleavage site is lost, it becomes more pathogenic. And he says he's interested in the exact role of furin cleavage sites. So from the horse's mouth, the person who added this protease cleavage site manipulation into the grant was specifically interested in furin cleavage sites. And he mentions this FIPV example, which was again at the S1, S2 location. Now, this is another piece of evidence and another coincidence. If we look at furin cleavage sites found in FIPV, we do find this PRRAR sequence, which is the same one found in SARS-CoV-2. Now, this one is actually starting at position P2. It's not adjacent to P1, so it's not quite the same, because the proper sequence should have the S directly after the R, so I'm less confident on this point, but it's still quite a coincidence that he's interested in FIPV and the exact furin cleavage site, the PRRAR addition, is found in FIPV. And this is pretty much the paper he's talking about, where the FIPV positive cats have mutations at S1 and S2 that deactivate the furin cleavage site. So this seems to be the paper Ralph was referring to, and PRRAR, just like we have in SARS-CoV-2. And it's also worth noting that we also find RRAR, the exact same cleavage site in SARS-CoV-2, in ENaC-A or ENaC alpha, which was being studied by research teams at the very same university that Ralph was working at. So he would, people at these universities, they talk to each other, they attend seminars. He would have known someone else was working on it. This furin cleavage site would have been known to Ralph Barrick. And while people say it's not canonical, if we consider they were talking about adding human-specific cleavage sites, this is a cleavage site found in humans, and I don't know why they would want it to be specific to humans as opposed to just compatible with humans. I'm not sure if this was a poor choice of words, but in fact we find it is fairly specific to humans. We only find this RRAR cleavage site in humans, chimps, and gorillas. It's not present in other mammals. So this is actually a human-specific cleavage site, or very close to specifically a human site, just chimps and gorillas within SARS-CoV-2. So at this point, we're at they're sampling where the relatives are found. It's spilled over where they were doing their spillover studies. It has the divergence target that they set, even though they had their logic for setting that target makes no sense. And it has a human-specific furin cleavage site like they were looking for, or like they wanted to add. These are quite a few coincidences already. And even more so, the evidence tells us that this furin cleavage site was recently acquired because there's a mutation that compensates for the addition, because the addition of furin cleavage site interferes with spike trafficking and has some interferes with the stability of spike. And this is a single nucleotide mutation required to compensate. And the lack of this mutation suggests that the FCS acquisition was very recent. And there are multiple papers saying that, and they, like here, it says the furin cleavage site insertion and these mutations likely occurred in quick succession. And since this mutation didn't occur until after it started spreading in humans, that tells us it started spreading in humans shortly after the furin cleavage site was inserted. So now we have a coincidence between when they proposed, in the timing between when they proposed insertion of a furin cleavage site and when this virus acquired a furin cleavage site. Now, I'd like to address some of the claims various virologists of questionable ethics and some of the claims they're making on social media. They'll claim it's only envisioning the use of pseudovirus, or that they would never use a new infectious backbone, they would use one that's already published. So it's worth noting here they have this nice little flow chart where they select the quasi-species with human infection potential and construct chimeric viruses. Not pseudoviruses, but chimeric viruses. And here they talk about synthesizing new genomes, consensus candidate genomes. Not simple chimeras, but using new genomes which would have a new backbone. And in the interview, Ralph Baric mentioned this, that they would first study in pseudotypes, but then they would move into chimeras and then virulent viruses. So any virologist claiming on social media or elsewhere that diffuse only would use pseudoviruses, or that they wouldn't use a new backbone, is simply not telling the truth. Now, I would also like to discuss this idea of them having new backbones. As to whether or not they were present in the database, you would have to actually look at the database. And here we see on September 12th, the database essentially went offline. Then it went sporadically online briefly in December. That doesn't mean it was accessible outside, but it was accessible to some researchers within China. Then it was offline again, sporadically online, and then definitively offline. So far, there's been no access to this database outside of China, so anyone telling you they didn't have the backbone is speaking from a condition of ignorance, because you don't know what they have. So just to summarize the coincidences, the Wuhan Institute of Virology proposed inserting these FCSs in 2018, and the first SARS coronavirus with an FCS spilled over in 2019. Did I say 2017? So in about a year after they proposed such a insertion, we see a coronavirus with such an insertion, and we know this insertion was recent and within that time frame based on this compensatory mutation being initially present. We know in 2018 they were actively seeking a virus with 10 to 25% spike divergence on very questionable logic, and in 2019 a virus with just that spike divergence spills over. We know the outbreak started sometime in September to November 2019, based on, in my previous presentation, and that's right when their sequence database goes on offline. So these are a lot of temporal coincidences. And then I would like to, I'm additionally drawing various adverse inferences, although I'm going to use the term beyond the legal terminology, which is simply where when you don't have the information and somebody refuses to provide it, you can draw an adverse inference as to what it might contain since they're not providing it. But I'm also drawing adverse inferences from when they basically do not tell the truth. What I would consider a lie, although that requires intent, and I don't know their intent. But we know they repeatedly withheld information that was subpoenaed, like in this testimony with Congress by Barrick. He's submitting. First off, there was this grant that was, the grant report was over 2 years late. They claim they tried to submit it in September 2019, but a technical error stopped them, even though there's no record and nobody else had this technical error. So this is already a BS explanation that I'm drawing an adverse inference from. The earliest draft we have is post-pandemic, or at least the earliest one they've provided is from 2020, which is still too late. And here they've changed the conclusion as to how rare these spillover events are. And yeah, they have not provided this information. And they had years to provide it. They still have not provided their early pre-pandemic drafts. They're making statements online, but just before the pandemic, about how they had all these new strains that bind to human cells, don't respond to monoclonal antibodies. And what are they talking about? This might refer to SHC-014, but he's speaking in the plural sense. So they're clearly not showing us some of their other strains. And in fact, they haven't, before the pandemic, they hadn't published any sequences collected after 2015. So we had years of gap as to what they have. And the fact that they wouldn't show us what they have, I'm drawing adverse inferences from. And I would also like to point out, we can look on Chinese databases, this grant database, where they're talking about 2 new SARS-associated coronaviruses, 2 new pathogenic ones in transgenic human mice. And this is by a researcher at the Wuhan Institute of Virology, and of course we never hear about this again because they're not sharing the data. I'm excluding evidence of reports of sick Wuhan Institute of Virology researchers because can't verify it. I'm excluding the intelligence community assessments like by the FBI because those can be shaped by politics and they're not showing their data. And I'm not going to discuss for the moment the restriction enzyme pattern matching the diffuse proposal because it's possible this showed up naturally. And there could be some cherry picking of enzymes used, although, and most recently a patent just came out where they were using those very same enzymes, so I don't think it's cherry picked. But for now, I won't get into that one. So we have, to recap the evidence, we have the place of spillover and where the spillover studies were conducted, the location of the relatives and where they were sampling, the timing of the spillover, and the timeline of the spillover study proposals, the spike divergence threshold and the spike divergence targets, the presence of a furin cleavage site and the proposed addition of a furin cleavage site, the actual specific sequence, this non-canonical one that's found only in humans and close relatives, and the FCS sequences they were interested in, the timing of when this FCS was acquired and the timing of their plans, and the timing of the outbreak and the timing of them taking their database offline. These 8 pieces of evidence all point to the lab. And together, I think they make a very strong argument that it likely came from the lab. So, in summary, where it spilled over, what spilled over, and when it spilled over. And as far as the evidence against the lab leak, the only evidence against it is that they didn't publish work on this prior to the outbreak, and there's a chance that they would have published at least a RDRP sequence like for RATG13. But we do have to keep in mind they haven't published any sample collected after 2015, or at least they hadn't before the pandemic came out. So this objection, I do think it reduces the odds of a lab leak being right, but I do not think it's enough to overcome the weight of the evidence. So they had motive, they had the means, presumably, we don't know what exact virus substrates they had, but they were clearly sampling in the right location. And they had the opportunity in the form of Chinese funding for that 2018 one on the two novel pathogenic ones in mice. And they had the funded grant from the NIH, the Understanding the Risk of Bat Coronavirus Emergence. Motive, means, and opportunity were all there. Now, I would also like to get into the false and misleading statements they've been making, which in my mind, when people very close to the lab start to make false statements that they should know are false or misleading statements, I'm drawing adverse inferences from this, and if the evidence was in their favor, they would just show the evidence. If they have to make false statements and misleading statements, to me that makes them look very guilty. So we can talk about from Shi Zhengli, when they first published on disclosed RATG13, they claimed they only found an RDRP match, and then they carried out sequence on the sample and showed a high match. This clearly implies they only had the short RDRP and they did the sequencing after the outbreak, otherwise they would have aligned the whole genome. But it turns out they actually had the whole genome in 2018. So they would have aligned the whole genome right away, not just this short RDRP. So it seems they're already misleading about when they had sequenced RATG13. And they also admitted that RATG13 came from a cave where 6 miners had been infected with something, and I think 3 or 4 of them died of pneumonia. Before the pandemic, in some PhD and master's theses, it was described as a viral pneumonia. After the pandemic, Shi Zhengli claims it was a fungal infection. And if it was simply a fungal infection, we have to question why, if these miners were sick in 2012 and the samples were sent to the Wuhan Institute of Virology, if it was just a fungal infection, why did they suddenly start going to this cave year after year after year? Because they did. And I do not buy for a moment that it was simply a fungal infection. So they're lying about when they sequenced it, or they initially lied about when, or misled about when they sequenced it. I think they're lying about those miners having simply a fungal infection. Yeah. And they did fail to disclose that RATG13 came from a cave where miners had been sickened with pneumonia and died. These are all very relevant data that they withheld. Then she claims they only had isolated 3 strains of SARS-related coronavirus. And yet, in the EcoHealth Alliance grant reports, they claim to have 11 isolates, including 4 previously isolated, for a total of 15. It's possible this grant was confusing genome sequences and isolates, but given their other statements, I'm uncertain either way. Their published data only supports sequences, not isolates, but they clearly said isolates, and these people should know the difference. EcoHealth Alliance has been claiming that no work was ever conducted in Laos. The emails don't show that they were sampling in Laos and sending the results to Wuhan. But yet they clearly were. Here we have Shi Zhengli and Daszak. Daszak is the CEO of EcoHealth Alliance, publishing sequences collected from Laos. Their grant reports showing sequences from Laos. Why are they lying? This makes them look very guilty in my mind. Then we have Eddie Holmes, a honorary professor at Chinese universities who has his name on some of the sequences that were collected by the Wuhan Institute of Virology, and was actually closely working with them just before the pandemic struck. And he claims they were only going to insert at S2, not S1, S2. And RRR is not from a human cove, and they were going to insert human-specific ones. Of course, this is an equivocation fallacy because we don't have, it's not a human coronavirus that needs to have a human-specific. It would just be a cleavage site that's only found in humans or specific to humans. So saying it's not found at a human cove is a red herring. Stuart Neal says, oh, it does not contain any proposed studies to add these. And then we have Anderson claiming that it would only be at S2, not S1, S2. And yet, if we look in the proposal, they've mentioned position 757 in other coronaviruses, which is the furin cleavage site. Within SARS-CoV, now this figure is a bit of a mess because it's using numbers from other the notation is inconsistent because the position numbers are different depending on if it's in MERS or SARS. But they mention other cleavage sites have been identified, 667 to 668, 678 to 679. Those are all at the S1, S2 boundary in SARS coronavirus. And these are all ones that they mentioned are cleavable by trypsin and capsaicin. And here in the grant, they mention mismatches that can be activated by trypsin or capsaicin. So these are S1, S2 sites in the grant activated by capsaicin or trypsin at S1, S2, furin cleavage. And furin is the only, they're mentioning furin cleavage sites. It's the only one they mention by name. They say proteolytic sites and furin cleavage sites. They don't even bother mentioning any other protease other than furin cleavage site by name, with the exception of exogenous trypsin or capsaicin. They are clearly talking in this grant about adding furin at the S1, S2 site. Anderson, Holmes, Neal, they're all misleading you. It's just, no. Well, why is this? Then he was claiming it was furin cleavage sites only because he said they would ablate sites, it would be pseudotype particles only because they mentioned ablating sites based on pseudotype particle studies. But this would be the pseudotype particle studies guide what they choose to ablate. They're mentioning introducing the changes into the parental strain. That's not a pseudotype. And if we look within their grant, in the early drafts, they mention a flowchart where they have testing, chimeric production, full-length recovery, and then predictive modeling. And that's what you see here. Select, they predict and screen with pseudoviruses, and that will guide selection for further characterization, which they then go on to with chimeric viruses. Chimeric viruses are used to select strains for full genome length recovery. And here they specifically want to test whether backbone genome sequence alters the spillover potential. You can't do this with pseudovirus. And this was described as a workflow or a pipeline. And after the full-length validation, then they go on to test their synthetic modifications. Their synthetic modifications come after they have the full-length virus. The idea that they're just doing this in pseudovirus is bullshit. So here's Christian Andersen lying. And here we have Ralph Baric himself, who wrote the proposal, saying they would build virulent viruses and then knock out the furin cleavage site or manipulate the cleavage site. This is simply not true again from them. It was not simply using pseudotypes. So prominent virologists are misleading the public about diffuse. Why are they doing this? When we look at all the evidence, it clearly points to a lab. When we look at their private communications, they were initially suspecting a lab. Then they are on Slack while having a phone call with Fauci. And they get a big ask. They want to hop on a call afterwards. And they say they have to pull something off that's hard to pull off. I think they've been asked to make it look like a zoonosis, and they think it's going to be hard to pull it off, to make it look like a zoonosis instead of a lab leak. And in my mind, they didn't pull it off.